Hello everyone. In this video I'd like to talk about PCR, which is more commonly known as the polymerase chain reaction. And this reaction is a method of copying fragments of DNA, especially small fragments of DNA, designed to get a larger quantity. So you can see it's a useful tool, particularly for DNA analysis. And that's why at crime scenes, if there's a small sample of DNA that the police can use, then they will use the polymerase chain reaction to gather a large amount of that sample, particularly if they're worried in case of contamination or anything like that. It should be said that this is an example of in vitro technology. Now, in vitro technology means working outside of a body, sort of like in a lab. It's in contrast to in vivo technology. So this is an in vitro technique. So we're just going to talk through what PCR is. So we've got a diagram here, and we're going to start essentially at this point here. We're going to look at this point here, because this machine that does this is called the Thermocycler. So it's in sort of three parts. So the first part is this separation of DNA strands. So this is the first part here, this yellow sort of part, third of this circle. So you have a small fragment of DNA, and at 95 degrees C, very specific temperature, those fragments get separated. So we break the hydrogen bonds between the bases, the A, T, C's and G letters in that DNA strand. We get two separate fragments. The machine then lowers to 55, and at 55 we get the addition of primers. So you can see you get these two sort of primer molecules there. Now the job of those primers is essentially to bind to the ends of the fragments, and that will help initiate new nucleotides being joined onto the DNA fragment. So just to be clear, a nucleotide is a sort of part of the purple strand that you see. So these purple strands here with the base attached. So the sort of little coloured part on this diagram, that's a nucleotide. It's actually the sugar, the phosphate and the base. But we're not going into that sort of technicality. We're just going to talk about the actual reaction. So we have these DNA fragments separated at 95 degrees C. Then we add these primers. We need two because we've got two fragments, and that happens at 55 degrees C. We then introduce, in the final stage of this reaction, an enzyme called DNA polymerase. So DNA polymerase attaches free nucleotides onto those strands. And what you can see is if you follow this round, we end up ultimately with two whole new fragments of DNA. So we started with one, we've simply split it, added free nucleotides onto both strands, and we've ended up with two. And this machine just repeats and repeats and repeats. It's a way of multiplying our quantity of DNA. So just a few things to consider when we look at PCR, a few important aspects. It is important that the fragments of DNA used are not contaminated with any other biological material. It's a common exam question. The reason why is that any other contaminant in this reaction would also be copied and obviously would make the results less reliable. Interestingly, this enzyme called polymerase comes from a particular bacteria, a bacteria called Thermos aquaticus. So I'll just write the name up here. Thermos aquaticus. And that's where the DNA polymerase comes from. So DNA polymerase enzyme comes from Thermos aquaticus. It's quite an interesting thing to note because many enzymes, like DNA polymerase for example, many enzymes have an optimum temperature they work at. Now, usually we say that's about 40 for many that we talk about in school, certainly. So it's quite rare to find one that is operating at 72 degrees. So clearly this bacteria must come from a specialised region, and it is. This Thermos aquaticus is found in areas like hot springs, so it's able to tolerate such high temperatures, and that's why the enzyme works best at that particular temperature. And it's important that this whole process is monitored, because clearly if 72 is the optimum, if we go very much below or beyond that, we could particularly denature that enzyme. And this, in a nutshell, is the whole of the polymerase chain reaction. So, stage 1, we take the DNA fragment and we separate it into strands. Stage 2, we add primers onto separate strands. And stage 3, we use DNA polymerase to add nucleotides onto both strands to make 2 new fragments. And we can just repeat this, so the 2 will then become 4, 4 will then become 8, etc. until we've got a desired quantity. And that ultimately is the polymerase chain reaction. Okay, I hope that helps.